Hello. So here's the lecture on prions. And there's our little mascot is a prion that's spinning around up there. Looks like a protein, right? It is a protein. Prions are protein-only infectious units, basically. So it's a thing that's a protein that can colonize another host like a virus would, but it's only just one protein, no genome whatsoever. And the scary thing, if you like, is that the protein is already inside you. Yeah, the killer's inside the house. Really cool. Huh? So this is a disease where you already have this protein, but your version of the protein is in a slightly different shape than the disease version of the protein. And it is that shape that determines whether it causes diseases. Now, the most common question that I get asked in Texas is this the thing that's in chronic wasting disease? And, when people have heard of prions, some of them have heard of mad cow disease, although the big outbreak of that was a while ago. It's back in the 90s. And so people forget. Yeah, all sorts of things. But this is a real thing. So chronic wasting disease, you can see up here, this poor little skinny animal. And this is something that you get in mule deer, and I believe you also get it in elk. It is an acquired condition, so it is transmitted to them. We're not sure how they are picking it up, although in mad cow disease, you would get this by eating proteins from an animal. And there have been cases where vegetarians also got this because stuff like gelatin can be made from animal hooves. It's just sort of boiled up animal hoof protein is one way to make it. There are vegan versions of gelatin that wouldn't come with this. And that was enough to get some transmission of the prion. So, the prion protein that you see above and these two that are over here, those are partial, but they're the part that we can get a good structure of. And so that's why that's the structure you will see over and over again. I believe they are missing the copper binding domain, at least part of that. What this does is, if you look at the arrows, they're pointing to little tiny things that you can't see. So I don't know why they even put them there. But if you look inside of the cells, you get these blobs of protein. And the blobs of protein are going to be surrounded by other proteins that are trying to break that down, trying and failing, because prion proteins are not indestructible, but pretty close to indestructible. They are really hard to get out. And when they bump, when the disease form of the protein bumps into a normal form of the protein, it turns into another version of another copy of the disease form of the protein. So it's like a meme. When the structure of the protein interacts with another one, then the information is passed on, even though there's no genetics involved. No DNA is changing hands. So prions, interesting. They behave a little bit like viruses in some of the stuff that they do, which we'll get into, but they are definitely not viruses, and they are not classified as viruses. But it's one of these things, where else are you going to talk about this? If not virology class, and it's in our syllabus. So here we go, prions. All right, this is from, I think it's the same paper as the one above. It's looking at transmission of prions. Neat experiment. So what they did was they got the sheep version of the prion. And sheep prions are, they cause a disease called scrapie. And in the chronic wasting disease, you'll see you get this sort of ruffled fur. They stop eating. In scrapie, you'll get some of the same things. It's a mental disorder. It's destroying their brains and central nervous system in general, I believe. And the sheep will start rubbing themselves, if I remember right, on fence posts and anything, but like too much, like until all the nice sheep wool comes off. And that's when they're really sick, and at that point, eventually they would have trouble standing because their nerves just don't work anymore, and they could potentially die. Yeah. So what they did in the experiment was they made some transgenic deer that had, or rather, they had some regular deer. That's up at the top, and they look like deer, so that's nice. And they had some transgenic mice where they put the deer version or the elk version of the prion protein in there to figure out, can we transmit from sheep to deer and then from deer to elk, Or can we transmit from sheep to elk? Yeah, because we know from previous work that it is possible to transmit the sheep prion to deer, and this is reinforcing that. And so when you go from sheep into deer, you get disease, but it takes almost 2 years, a little over a year and a half, to actually get that disease. Thing is, when you take it into the mouse that makes a nice amount of this prion protein, that changes somehow. Probably there is a small structural change, some small but important change in the disease form of the protein. And now, when that goes back into deer, it only takes 10 or 11 months, so less than 1 year, to cause disease. So basically, you're cutting the time until disease in half by running this thing through a mouse with deer proteins before you run it through a deer. And that's weird. What they also found, though, was that you can't really get good transmission to mice with the elk version of that protein, the prion protein. And even if you run it through deer first, then you can get transmission to the elk. So deer can be a gateway, just like think back to COVID. Yeah, so humans and bats probably never in the same place, but humans and other animals all the time in the same place. And so if you get bats and other animals, and it gets transmitted to the animals, and then the animals are with other animals, and then you get more and more and more, and then eventually people are there and they're buying and selling the animals, then that's a great way to end up going from bat to human with a couple intermediate steps, and then you get things like COVID. Yeah. And so it's the same thing here. You go through a deer, and now you have a prion that is able to spread to elk as well, when the original one wasn't. So that's terrible. But yeah, also that's the truth. And so yeah, you can see here we go. All the mice eventually do get infected, but the deer prion protein mice got infected sooner, yeah, than the elk prion protein mice. All right, so if you're not a big time, big game hunter with all those, buck heads on your wall or whatever, here we go. It's a little story about, I wouldn't call it head hunting, but it is human cannibalism. So in Papua New Guinea, in the 20th century, actually, so here's data, and there's a disease that came up, and it was specific to human cannibalism. And so I am not the ethnologist that you may think I am. So the broad strokes are that you got different groups here. They're related to each other, and groups are always fighting. Yeah, I don't think that's anything that's new or specific to this particular area. In this case, the fights would sometimes result in deaths. And one of the things that happens after killing an enemy is that some of the people would consume cannibalistically parts of the enemy, including the enemy's brain. Brains are full of prion protein, and so if the brain has a prion protein that is the disease shape of the prion protein, and I'll show you what that means in a minute, then you're potentially transmitting a lot of that to the person that eats the brain meat. So if you were, thinking, well, is there a downside to human cannibalism? Yes, this is the downside. There's the potential to spread this. The disease is called Kuru, and you can see here there was a real push to kind of stop the practice of human killing and eating. To stamp that out, which, whatever. We do things that are traditional too that make no sense whatsoever. So yeah, it's not, not an us versus them thing. It's just here's the consequence thing. And you can see that it was going strong for a while, and then eventually it did sort of taper off. 61 to 63 is when they really clamped down on it. And there were not many cases. Only the white parts of the bars are from 61 to 63. So there you go. And it's very rare today, thankfully. But we know that's a thing. Consuming human brains, if they have the wrong form of the protein, does seem to lead to transmission. And we know from the bovine version of it, which caused this thing called variant Creutzfeldt-Jakob or Jakob-Creutzfeldt disease. We know that that version can survive some heating. So even if you have cooked meat, but potentially it's not completely thoroughly, cooked to the point of being well done, where people don't like to eat meat anymore, there is at least some potential that if the disease form of the prion was in there, that it then survives going through your digestive tract, which that's a tough way to go, makes it somehow into your bloodstream, travels through your bloodstream, makes it to your brain or central nervous system, bumps into more of these things, and then something like a year down the road or 2 years down the road, then you get this debilitating disease. We'll show you a picture at the end of what a human brain with new variant CJD, I would normally switch those two around, looks like, and it's pretty bad. It's like the pathology would look a little bit like Alzheimer's, in that this is caused by a sort of toxic buildup of more or less indestructible protein bundles in the brain and the malfunctions that happen as a result of that. All right, so here's our little thing. This is all our different ways to be a virus. And then there's this guy. Yeah, Stanley Prusiner got the Nobel Prize for essentially discovering, or I don't know, he did some of the important work showing on how prions work, how they are transmitted. And so that's why he's up here instead of David Baltimore, who did the old classification based on which of these replication methods you use. And now we use the different classification that's loosely based on this, but not that closely. It's not as strict as the old version. In terms of, if you have an RNA genome that's single-stranded, you have to be in a certain group. Actually, that part's still there, but the other versions are a little different. All right, so prions bypass this whole genetic thing. You don't have RNA, you don't have DNA, it doesn't replicate via anything. You just have proteins, and those proteins are going to bump into other proteins that already exist. And if those other proteins don't exist, then you can't transmit the evil protein shape, which is what a prion is. Yeah, shape kills, I guess. Is that a moral? I don't think that's a moral. Here is a look at what the prion protein may do. And this is a reasonable guess as to what the prion protein looks like. So you can see these green things over here, and apologies if you're colorblind, they're the things that are little coils running on the back. And you can see this pink thing here. Yeah, not the friendliest set of colors that they picked. This is what's called a beta helix. These are what's called alpha helix. Alpha helix is the normal version of the helix, where you're going to have 2 turns every 7 amino acids, I think. Is that right? Yeah, that's right. I think that's right. And with a beta helix, they're kind of bigger, longer things, and they're going to be held together by the same sorts of interactions. It's like 3 beta strands, and over again, they're going to be sort of folded up into a sort of a helix type shape, it's like a Toblerone. If you've ever been to an airport, those giant triangular chocolate bars that are spiky but delicious. Yeah, I guess. So this is the healthy form. This is the disease form. We're going to call the healthy form the PRPC, prion protein cellular version. We're going to call the disease form the PRPSC. The SC stands for scrapie. Scrapie is the sheep version of this disease. And we're going to call it the PRPSC disease form, whether it's human or sheep or cattle or deer or a mouse with the genetic prion protein of an elk. Yeah, it doesn't matter. PRPSC. What is this prion protein that you've heard so much about? Well, it's a neat little protein. In the N-terminus here, you've got these repeats, and you'll have different numbers of these repeats. The repeat is going to be an octapeptide, which means 8 amino acids, and they're going to repeat over and over again. The repeats, I believe, are the thing that folds up into the shape, because if one of them can make that shape, and you have more than one of them there, then you're going to get more than one of those shapes. Yeah, for sure. This is the globular structure. The repeats seem to influence this. It is actually this part, I believe, that is thought to be the one that transmits the disease, but the repeats do seem to influence this as well. And you can tell this in just in with purified protein. So here are the different versions. So this is prion protein where they take out the octopeptides, and you can get some little sort of protein fibrils. So this is a protein that would normally be loose, and loose protein is going to look like this, just little sort of blobs on the background. But if it's forming up into the prion form, you're going to get these long sort of chains of helices. The helices will stack together, and yeah, you get these little sort of hairs. And then, okay, what if we put in one octopeptide repeat? Ooh, you get more of them. What if we put in 5 octopeptide repeats? Yikes, look at all of those. What if we put in 8? And yeah, look at that. You just get these sort of clumps. So the number of octopeptide repeats is going to determine how likely this thing is to form up into the disease form if it is exposed to some of the disease form. And this is a neat little assay that you can do. You just sort of shake it at 37 degrees, and the proteins form up into these fibrils all by themselves. Yeah. So it's a thing the proteins could do, probably as an accident, maybe related to their function. Hard to tell, because we don't have all the information on the function of this protein. There are still some weird gaps for sure. Here is a generated version and 3D structures of the sort of an array of the disease form of the protein. And so you can see that. Look at that beta helix right there. Ooh, looking good. And so they're going to pack, this particular version is going to pack in groups of 3. I don't know how many octopeptide repeats this particular version has, but they look great. They stack up, and once you make one of these things, then it can stabilize 2 others, and so that's probably what is making this fibril. It's just these 3 things next to each other, and they're sort of stacked units of 3, like little triangles of triangles extending out into the distance. Okay, so tell us more about the protein itself, because why not? Yeah. So prion protein cell associated, here you go. You got a flexible N-terminal region, looking great. This thing is glycosylated, so here's our glycan structure. And it's a protein that has a thing called a GPI anchor, which is a glycosylphosphatidylinositol. Yeah, there we go. So this thing doesn't have a transmembrane. It's not the protein itself that's going to go through the plasma membrane. It's going to be covalently linked via the C-terminus to a lipid molecule. So like there's a lipid grafted onto the end of this protein. And so the lipid's going to sit in the membrane like all the other lipids, and the protein part's going to hang out next to the lipid because they're covalently attached. And that's how that works, yeah. Bonded for life, yeah. There would be ways inside the cell, enzymes that would break this bond. And there are enzymes, of course, that make this bond. So there are several versions of this thing that exist. The PRPC would normally have 5 of these octopeptide repeats, I believe is what you're seeing here. Alpha 2 and alpha 3, these are the big long alpha helices that are over here. The beta 1, beta 2, and alpha 1, these are the little bits. Here's your tiny, they're little blue right there. You can just barely see them. Yeah, those little things. Those are your beta strands, and so those are either side of this short alpha helix, which you've got right there. And that's the part that is going to be involved in the disease process and the conversion to disease process. That's like the thing that starts, and then if you have these octopeptide repeats, then those will then extend the fibril formation. Yeah, so kind of neat. And so there's a signal peptide on here, and looks like also a C-terminal signal peptide. There's a G, and this is the signal peptide that leads to GPI anchoring. This is the signal peptide that leads to exporting up to the surface of the cell. So it's got a ticket for both shows, and it's going to go to both. That's the idea. There's a disulfide bond that holds between alpha helix 2 and 3 together. And the glycans are attached, one to alpha helix 2 and one between those two alpha helices. I believe that's the one that's sticking up here, the big one. These are some related proteins called Shadu and Doppel. Yeah. And so the prion protein looks like a combination of these two. So it's got the C terminus of something like Doppel, and it's got the N terminus of something like Shadu. And if you imagine sometime back in history, these two things getting recombined and getting put together in somebody's genome, or some creature's genome, they're in all kinds of animals, and even in yeast, then that may be the genetic event that made all of this possible. Yeah. There are other protein folding diseases. That's like a whole thing. We're just talking about the prion type of protein folding diseases, because most protein folding diseases are going to be inherited. They're not going to be infectious like that. So where do you get this cool PRP prion protein? The gene is the PRNP gene. Where do you get it expressed? Well, a lot of places that are all in the brain. So here's go. A ton of it's in prefrontal cortex. The amygdala is loaded. Spinal cord's got a pretty good level. And so there you can see. Other parts of the body, not so much. A couple of your glands have a little bit. But look at the colon. Ooh. So the colon happens to have an unusually high amount of prion protein expressed in it. Why? I am not sure. I'm not sure what its function is in the colon, if that is even understood. The papers that I've read did not have a great explanation for that. But it's there, and so that gives you exposure to the stuff that people eat. And that means that there is a chance that you transmit not the protein itself, but the structure of the protein misfolds and causes misfolding of the version of the protein that's on the colon cells. And if those somehow have a way of getting cut off and floating through the blood, that's how you get the transmission. That would be a path that would make a lot of sense. Not worked out. And then you can see most other tissues, like all these sort of big important tissues, very low levels, but not zero. Yeah. So there are a lot of ways for this thing to potentially bump into another one and bump into enough of these things. And look at that, uterus. Yeah, yikes. Which would mean that would be a possible means of transmission. Possibly even to the unborn. Yeah, which would be terrible. So here's your prion proteins. They're going to get processed. There are atom family proteases that are actually going to cut them just above where that GPI anchor is, and that can then send these things off, and they will then be soluble. The N-terminal region can then sort of misfold and potentially bump into other proteins that are still anchored on cell surfaces. Or these things may all meet up outside of cells. Cells may then try to sort of swallow and digest the entire blob of these things. This is our current understanding. So new protein is going to come out via the ER to the Golgi to secretory vesicles up to the surface, just like any other surface expressed protein. It's going to be modified probably in the ER, I think is where all those anchoring modifications are going to happen, and the early steps to the glycosylation. All right, what does prion protein do? A lot of things. This is the older understanding, and it's based on, well, what if we knock this thing out? And so if you take something like a mouse and you knock out prion protein, so there's no prion protein, 0/0, there you go, then what you get is mild disease, and it seems to have a little, the idea is that it has a little bit of its activity, but it's a thing where you need to have the activity binding to, or rather, this is the prion protein down here. The idea is that there is a protein that it binds to, and there are a lot of proteins that it binds to that we know now, but we don't know for sure which ones are definitely associated with disease. It's a little more nuanced than that. We'll get to that, I think, in the next slide. But wait for it. So if you have the prion protein interacting with the other marker or markers, it could interact with a bunch of things, and we know that it does. Then you're going to get some kind of signal, and that signal will basically cause the central nervous system to not destroy itself. But if you have no prion protein there, then you'll get less signal through here, and you do get pathology. And the pathology can involve basically learning difficulties in the mice. Put in a broken version of this, or a stabilized broken version where you've chopped out part of the protein, and what you'll see is that you get some interaction potentially with these other proteins, but you get maybe none of the signaling coming through, and then you get extremely severe pathology. This is like an understanding of why. So it's not the prion protein, it's something the prion protein would be interacting with that is going to cause the bad thing to happen. And there are ways to stabilize this and make more of an interaction possible that lead to the most severe pathology. In the worst cases, you're going to get essentially destruction of the insulation around the nerves, the myelin sheath. Yeah. In addition to all kinds of horrible, brain problems. So, prion protein is going to signal through a lot of different co-receptors, and this is going to signal through some of the giant signaling pathways. So cyclic AMP, protein kinase A, PI3 kinase, AKT, ERK1 and 2 signaling, and RhoA or ROC, as well as some protein kinase C and some SF kinase signaling. And those have that's like a list of the most important signaling mechanisms inside of any cell. So it looks as though there are a lot of things that are at least reportedly modulated. And again, we know a lot of things that this protein can do. We don't know which ones specifically in sort of what proportion are leading to the disease, but this is like a start toward getting there. And in a year or two, we'll probably have a much better picture. Here are some of the proteins that are reported to interact with PRPC. So it definitely interacts with a lot of stuff. And this is showing the method that they determined, like co-IP is if you have an antibody against prion protein, it'll also bring down the other protein, or if you have an antibody against the other protein, it'll also bring prion protein with it because they're stuck together. Yeast 2 hybrid. There's a lot of methods for telling if two things are sticking together at least for a little while. So any of these things, all these things, none of these things could be involved. And some of the ones that aren't directly involved in disease could be involved in transmission. Because somehow, remember, this protein is going to get from the gut up to the brain, and your gut cells are not allowed to go up there. So the protein's got to come loose, and it's got to interact with something. And it may be a case where it would interact with like a vimentin or something, and or interact with a laminin, something that's going to be outside the cell, and that'll get a little closer. And then once it's closer, maybe it can interact with 14-3-3s are usually in the cytoplasm, but there are going to be some of these surface, like there you go, your nicotinic acetylcholine receptor, which is a thing you would find on certain classes of nerves, and it would be exposed on the surface. And then if you get somehow transmission of either the protein into the cell or interaction of the signal, like if you have a version of the protein on the cell and you bump into it, boom, and now it's converted. Proteins on the surface of the cell get pulled back inside the cell all the time. Now, I don't know how that leads to this protein being in the right place to interact with other proteins or cause the disease, but hey, maybe. I don't know. Or it could all happen on the surface of the cell. A lot of unknowns with prion disease, but a lot of knowns. Yeah, I don't want to sell this as just, you can write down dunno and it's a good answer, because it's not. Yeah. Don't be lazy. Yeah. Or I don't know. You're not lazy, but there's no shortcuts in virology or prionology. Yeah. So there are different strains of prions. and depending on where you've got it from, there are differences in genetics, and there are differences that we think in structure. So some of the, if you make fewer or more, and we don't know which it is, of these beta helices, it may be more or less transmissible. And so that would be something like a virus, comes in different variants. So, all right, that's neat. You see adaptation with prions, where when you put it through an animal multiple times, it will sometimes accelerate. And so it'll take half as long to get disease, like we saw with the deer in the first two slides. And so that seems like adaptation, but it's not a genetic adaptation because the gene that codes for the prion is always part of the human genome and every one of these other genomes. That does not change. It is not modified in any way that we understand, or that I understand anyway. You can classify them by trying to digest them with a protease, which is going to snip them into the parts. And so you can see there are actually different protease sensitivities of these things. The proteinase K, is a really tough protease, but even then it can only cut like the complete thing into two parts, something like that. The amyloid form, the fibril form, is very, very resistant to cutting by any protease. And proteinase K is not a human protease, and our proteases are not even as good as this. So we really struggle to get rid of this thing. There are species barriers in that it can be transmitted to some species but not others. And if you go into one other species that may or may not have disease, you can then have like an asymptomatic carrier, and you can then get out the prion protein version, feed it to another animal or inject it in another animal, and get reproducible disease. So it can, skip generations and prions, So all of this stuff is stuff that would be like the stuff that viruses can do, but we don't define viruses by, what they do. We used to 200 years ago. It's like a tiny thing that makes you sick. But now we say, oh no, bacteria are different from viruses. Viruses are different from prions. And there's a lot of other disease-causing things in the world, chemicals, genetic conditions. So anyway, here's your brain on prions. And if you were to zoom into this, you would see sort of tangles. You would see destroyed brain matter. Look how small. Look at the gaps that you get here. There's just a lot of destruction of gray matter in the brain. Essentially. So protein-only infectious agents, they can convert other versions of this that you already have. You're expressing prion protein right now in your central nervous system. If you run into the wrong prion and it transmits its little evil structure up the chain from your intestines to your head, or if you inject yourself like a fool with prions, then it causes this. And it's pretty rapid toward the end, and erratic behavior. You'll have episodes potentially like you would get in mad cow disease, where the mad cow has trouble standing up. It's like a downer cow. And that's why you're not allowed to sell those, because there is at least a chance that those things actually have transmissible prion protein in them. Now, do you have an asymptomatic cow? Do you have a cow that got infected a month ago but isn't showing any symptoms yet? Those go straight into the food supply, the food chain. So, burgers. I don't know. Are they worth it? They taste pretty good, but There's a downside, just as with human cannibalism. There's a downside to all these forms of carnivorous activity. There's stuff that kills vegetarians too. Don't worry about that. But this is a special thing. So there you go. Hope you got something interesting out of that. Prions are kind of neat. And definitely the world needs to know more about how these things work. Maybe that'll be you someday doing this. All right. Thanks very much.